To continue with the lecture series on cell, we have already learned about prokaryotic cells and even eukaryotic cells. We talked about the cell membrane and cell wall. Today we will move into the structures that are located inside the cell. Now, first and foremost, we need to talk about endomembrane systems. Now, what are endomembrane systems within the cell, inside the cell? So, let us talk about it. Endomembrane systems includes those organelles or cell organelles, that is the structures inside the cell whose functions are coordinated. Means there are four such cell organelles, four such cell structures inside the cell in the cytoplasm, located within the cytoplasm, whose functions are coordinated. Like if one of the organelle prepares something, it will be transported to the next will again do something with it. So there, all the functions, they are coordinated, they are dependent upon each other. Such a system of organelles are called as endomembrane system. As I've already written here, organelles whose functions are coordinated, they are called as endomembrane system within the cell. And it includes four cell organelles: endoplasmic reticulum, Golgi bodies, lysosomes, and vacuoles. So four organelles are included within the endomembrane system. We include them under endomembrane system because their functions are coordinated. Now let us see a little more details about it. This is the structure of a cell. This is the structure of a cell, trying to show a three-dimensional view. So here you have the cell membrane, this white boundary here, that is the cell membrane, and then you have the nucleus in the center. Now we are not talking about that. We have already discussed what is the cell membrane, and now we are going into these structures located within the cell. Now endomembrane system includes endoplasmic reticulum. It includes Golgi apparatus. Here also endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. Vacuoles, this is an animal cell that has been shown here. Vacuoles are more prominent in plant cells. So even the vacuoles, they coordinate with this rest of the three organelles. Endoplasmic reticulum, here, this is the endoplasmic reticulum. Golgi bodies, they are always located quite close to endoplasmic reticulum. Lysosomes, which will be again here, and even vacuoles, they will be present. So now let us try to understand more about these cell organelles in details. First to talk about is endoplasmic reticulum. Endoplasmic reticulum are membrane-like structures. They are membrane-like structures folded upon itself, which are known as cisternal sacs. They are called as cisternae. Now they are called as folded structures as cisternae. They are continuation of the nuclear envelope, the membrane which forms which covers the nucleus in eukaryotic cells. The outer layer will continue to become the endoplasmic reticulum. They are, like you can see, the membranes, these are interconnected. So whatever materials are produced here, they can be transported. So main function of endoplasmic reticulum is usually it is transport of substances. They provide a connection with the nucleus and the outer, that is the cytoplasm. The endoplasmic reticulum, they may contain ribosomes. The type of cell organelle, ribosome may be present upon the endoplasmic reticulum. Then it will be called as rough endoplasmic reticulum, and if they are devoid of the ribosome, it will be called as smooth endoplasmic reticulum. Accordingly, the functions. The rough endoplasmic reticulum contains ribosomes, so their function will be to produce proteins. Translation, protein synthesis. Smooth endoplasmic reticulum, they are devoid of ribosomes. Their function will be the synthesis of lipids. So two different functions are associated with RER, rough endoplasmic reticulum, and SER, smooth endoplasmic reticulum. Now here we have rough endoplasmic reticulum, whose function is modifies proteins. okay, that is formed by the ribosomes, okay, that will be formed by the ribosomes which are present over the endoplasmic reticulum. And once the protein is formed, what will the endoplasmic reticulum do? They will modify that protein and produce tiny vesicles, globules. Tiny globules will be produced. The proteins will be converted to tiny globules so that they are transported outside. Smooth endoplasmic reticulum function is to synthesize lipids such as fatty acids, phospholipids, steroids. So they will be produced within the smooth endoplasmic reticulum, and then accordingly they will be transported into the cytoplasm so that they can carry out their own function. Golgi bodies. The next organelle, Golgi bodies. Now, once the proteins are produced in the rough endoplasmic reticulum and lipids also are produced in the smooth endoplasmic reticulum, after that they have to be transported to different parts of the cell. That transportation work, that further modifying it and then transporting it to different parts of the cell is done by Golgi bodies. What are they made up of? Even they are made up of a system of membranes. Membranes which are folded, interconnecting, which are they are also called as cisternae. In plants, Golgi bodies are also known as dictyosomes. So here is the picture of a Golgi body. Now Golgi bodies, here you can see they are folded. They are folded upon each other. They are folded upon each other, and always there are two faces. That means two main surfaces. One is called as the cis phase or the receiving phase, or also called as forming phase. And the other side is the trans phase, or which is the transportation or delivery phase. So now what happens, the protein structures in the form of globules or the lipids, they will be transported to the cis phase of the Golgi bodies from the endoplasmic reticulum. Then as it moves, as it passes one after another along the membrane, finally it is released out. Here you can see in the form of some tiny vesicles or tiny bubble like structures, they are released out, which we call as secretory vesicles. So they will be released, they will continuously emerge or erupt out of the trans phase. So their function, and during this movement, what has happened to the protein? It has got further modified. So that the other parts of the cell can use it. The space inside each of the bag-like structures, they are called as lumen. That is the empty space inside the Golgi bodies. Now the function, as already I have mentioned, modifies proteins and lipids, process materials to be removed from the cell. If anything has to be removed, that also processing will be done in the Golgi bodies. It secretes mucus and other enzymes, hormones, everything it secretes then for transportation. Then Golgi bodies, furthermore, here we have two more structures. This is 2D and this is the 3D structure. So when we draw, usually we represent it in this manner. The cis phase and the trans phase. Through the cis phase, the proteins will enter, lipids will enter, and to be delivered out through the trans phase. So here you can see the ribosomes upon the endoplasmic reticulum. They are producing the proteins here in the form of vesicles. They are released out. This is the transport vesicle released out by the endoplasmic reticulum, which reaches the trans phase, and as it moves, it is getting further modified, and finally liberated out through the trans phase. It's finally liberated out through the trans phase. If required, it will be used in the cell, or if it has to be transported out of the cell, see, through the plasma membrane, it can be moved out of the cell to be used by some other parts. So this is the main function of Golgi bodies. Next is lysosome, the next unit or organelle on the endomembrane system. Lysosomes are small spherical organelles which enclose digestive enzymes. Now, what are these digestive enzymes? These are also made up of proteins produced by endoplasmic reticulum. They're produced by endoplasmic reticulum, and then packaging is done by the Golgi bodies in the form of tiny secretory vesicles, which will be released out from the trans phase of the Golgi bodies. Then what is the function? The function is it will help in digestion of molecules. Lysosomes are found both in animal and plant cells. The main role of lysosomes is to remove the unwanted particles that will be present in the cell. Like it will remove any cell organelle which has died or which has got worn out. So such substances will be engulfed by the lysosome, will be taken in by the lysosome, and they contain enzymes which is capable of breaking down the particles. They can digest, they can break it down to the smallest possible unit and thereby totally dissolve that unwanted particle inside the cell. So also we can say lysosomes act somewhat like a garbage collector, digesting, breaking down the garbage of the cell. Why is it possible? Because they contain enzymes, hydrolases, mainly hydrolases, proteases, they contain those enzymes. Those enzymes are capable of breaking down any organic material. But sometimes, if the lysosome for any reason thereof happens to break down, then what will happen? All the enzymes will be scattered within the cell and thus it will start breaking down the cell parts. It might lead to even death of the cell. So as such, lysosomes are also sometimes called as suicide bags. So here we have the details: bags of digestive enzymes. Then so why they are membrane bound, as you can see here, so that the cell does not digest itself. But if for some reason the membrane breaks open, then the cell may get broken down. Then it breaks down any unwanted particle. Okay, it destroys bacteria, it can recycle damaged organelles. So these are some of the functions of lysosomes. Then now we can see here the coordinated role. The rough endoplasmic reticulum will produce protein. Transport vesicles will carry that protein to Golgi bodies. Golgi bodies will further process it and release out in the form of these lysosome-like vesicles. Even the lysosomes, they arise from the Golgi bodies. So now what will happen? The lysosomes, they can engulf the some foreign particle or damaged organelle, okay, and thereby see everything has got digested. So they can also, what can they do, like say an unwanted or say food particle has entered the food vacuole, also it will become the food vacuole. That means the food will be engulfed within it, within a membrane, and that will be digested by the lysosome. So lysosome, the main role is digestion or breakdown of organic material inside the cell. Why is it possible? Due to presence of hydrolases, that is the hydrolytic enzymes. Now, the last of the endomembrane system, vacuoles. Vacuoles which are exclusively present only in plant cells, large prominent vacuoles. In animals, vacuoles are present, but they are very, very insignificant or very tiny in size. Now, the vacuoles, they always have a covering which is called as tonoplast, which is the outer covering of the vacuole. It contains a cell sap. Cell sap. Now, what is this cell sap? So, vacuoles can be different types. If it contains water, we will call it as water vacuole, cell sap. Then, or if it contains some minerals along with the water, okay, it can also contain some minerals along with the water enclosed by the tonoplast, the outer covering. So vacuoles can be of different types. It can be gas vacuoles even if it just contains gas or air. So where is the vacuole located? They are always present in the cytoplasm. Okay, there is no specific place. Okay, like in plant cells, the vacuole occupies almost the entire space within the cell. The entire space is almost occupied within the cell. And in animals, they can have many tiny vacuoles if present. Now here again, and the difference of plant and animal cell vacuoles. In animal cells, tiny, as I've been mentioning. In plant cells, large. Then what is the function here? Mainly storage. It contains nutrients, water or waste matter. And plant cells, they will store water. Mainly, it is used to store water with minerals, which will help in pushing against the cell wall and thereby maintain the shape of the cell and keep the plant rigid, plant cells rigid, turgid. It will keep them inflated, turgid, to maintain the shape of the cell. Usually, vacuoles are helpful, or vacuoles will help. So we saw today the endomembrane system, which is made up of mainly the endoplasmic reticulum, the Golgi bodies, the lysosomes, the lysosomes and the vacuole. Endoplasmic reticulum will produce proteins or fats depending upon whether it is rough or smooth endoplasmic reticulum, that will be delivered to the Golgi bodies. They will pack it, packaging will be done, and then it will be released out in the form of lysosomes or other secretory vesicles that will be utilized by the cell, and if any excess components are present, that can be stored within the vacuoles. So that is about the endomembrane system. Next lecture we will go into the double membrane structure, the double membrane cell organelles. We will be discussing in the next lecture. So if any queries arises in your mind while going through the lecture, then you can freely post in the comment section. Thank you.